In this lecture, I'm going to briefly review the major developments over that period of time, talk about the revolution in single-cell genomics, which is advancing pre-implantation genetics, and then speculate a little on how pre-implantation genetics may become central to the practice of IVF. Over the past 30 years, there were really three major developments that stimulated the development of single-cell genetics enabling PGD. The first of these was the discovery, or some people would say the rediscovery, of the polymerase chain reaction. This really, for the very first time, made it possible to contemplate genetic analysis at the single-cell level. So prior to this time, making a genetic diagnosis on an early embryo, which has a maximum of a few hundred cells, would have been impossible. The second major development was the sequencing of the first draft of the human genome in 2003, 50 years after the discovery of the double helix structure of DNA. And that enormously advanced our knowledge of the DNA sequence, giving us access to hundreds of thousands of markers, polymorphic markers across the genome, which have really facilitated the development of PGD. And finally, and perhaps most excitingly at the moment, is the achievement of this remarkable achievement of the $1,000 genome. That is to say, we can now sequence someone's entire genome for as little as $1,000. And that was announced by Illumina in January 2014. And this has opened up the prospects, the real prospects of personalized medicine, and will also revolutionize pre-implantation genetics. So I don't have time to go through all of the developments, obviously, of the last 30 years, just to point out how rapidly this field has actually developed. So single-cell genetics began really only a few years following the first description of the polymerase chain reaction. And although the first animal models used a very sensitive biochemical assay, we very, very rapidly developed PCR protocols that enabled us to amplify, repeat, and even unique sequences. And so that within a space of only 3 years, we were able to make the first successful attempts at pre-implantation genetic diagnosis itself, as we'll see. We then moved on to techniques such as fluorescent in situ hybridization to look at chromosomes in an early embryo, and that showed us, at least gave us an inkling of the very high incidence of chromosomal abnormalities. And people began to think that perhaps we should screen for these to avoid transferring non-viable embryos. And so the first attempts and the first births occurred as early as the mid-90s, in 1994, and has continued since then, despite adverse randomized controlled trials. But we've really only had commercially available PGD and PGS tests for a few years, starting in 2008, as we'll see, with array CGH platforms that now enable us to have much more control and much more accuracy of the tests we carry out. So the first live births were actually in a series of couples that were at risk of X-linked diseases. These are diseases that typically involve gene defects on the X chromosome, and because males only have a single X chromosome, tend to affect only males, although some females can be mildly affected. And that was the case with these women. And what you see here is that the challenge was not only to diagnose the defect in a single cell, but also to do it very rapidly. So in those days, the optimal time for a transfer rate would be day 3 following insemination. And so we literally had 8 hours in which to biopsy the embryos and test them, diagnose them, get the patient back, and do the transfer if we wanted to get the patient pregnant. And what you can see here, that to do this, we used an amplification technique in this case to amplify a Y-specific sequence, a repeat sequence on the Y chromosome to identify a male in this case, and by its absence a female. And these are actually the 2 sets of embryos that were transferred in the 2 successful pregnancies. And these led to twin pregnancies. So what was remarkable about this early experience was that in these fertile couples, the implantation rate was actually very high. And from memory, it was, in the first 5 cases, was actually 45% per embryo transferred. The first twins to be born are in the top right-hand corner, Natalie and Danielle. And unfortunately, there was a stillbirth in the second set of twins. But this is the surviving twin, Rachel, with Robert Winston, my clinical collaborator, now Lord Winston. And this is me in the background, if you don't recognize me, when I had more color in my hair at that age. Since then, we've developed many protocols on the back of the effort to sequence the genome. And so now we can effectively do a whole range of single gene defects with specific protocols. So in 1990, we simply identified the sex of the embryo and transferred females that wouldn't be affected. But now we have specific protocols for things like Duchenne muscular dystrophy. And because we have the entire genomic sequence, we can essentially tackle any single gene defect. In the UK, this has quickly moved on to considering other conditions than serious childhood conditions. These include late onset conditions like Huntington's predominantly, and even cancer predispositions, BRCA1 predisposing to breast cancer. But the first one to be done was in fact achieved in 1998, and that was colon cancer, familial polyposis coli. Because we discovered that there were a lot of chromosomal abnormalities in early human embryos, this became an area of intense interest. And so very rapidly we developed protocols, as we will see, to try and identify embryos that essentially are not viable because they have a significant genetic imbalance. And one of the great achievements of one of the other pioneers of PGD, finally, was the development of techniques which enabled us to actually identify embryos that were HLA matched to an existing sick child. And in that case, not only can we avoid the disease in that sick child in the offspring, the baby, but we also have the possibility of collecting cord blood and transferring those cells to the affected individual. And now many children have been cured of blood-borne conditions in particular. This will be familiar to anyone that is involved in PGD. This is has been the current practice. So following the whole genome sequencing, it gave us access to these markers. One particularly useful set of markers, short tandem repeat markers, can be used essentially to fingerprint a chromosomal region that carries a gene defect. Now, I don't have time to go into all the details of this, but essentially this allows you to identify both the chromosome with the affected gene and the normal chromosome, and gives you much more accuracy than just simply looking for the single gene defect itself, the actual mutation. And what you can see here is a very nice example that was done in Francesco Fiorentino's lab, in which a multiplex fluorescent PCR reaction was actually used to look at the size of these polymorphic markers across chromosome 6 for the HLA region and also chromosome 11 around the beta globin gene in a case in which they were attempting to match, first of all, avoid beta thalassemia, but also to match. And so you can see here in general that the affected child here is carrying these pink chromosomes, in which case these embryos here are carriers, these are normal, and that's an affected embryo. But you can see that there's also a match here, a perfect match with the affected child. And so these 3 embryos could be transferred. Single-cell genomics is revolutionizing our approach, and this really began approximately 10 years ago when we and others had early access to a new form of polymerase, actually a polymerase isolated from a bacteriophage, which allows us remarkably from a single cell to amplify enough DNA to get micrograms of DNA, the same amount of DNA you'd get from a Corrion villus sample or a tissue sample. And this really changes the game, if you like, because you can now use conventional genetics in a conventional genetics lab And so already we were dreaming, if you like, about using genome-wide platforms, using this DNA, to overcome what are still today the deficiencies. So I should say we haven't overcome all of the problems, because still, when we amplify the whole genome, there are gaps. And we'll see that the strategies that we now use the power of genomics to actually get round those technical issues and nevertheless give us very accurate and powerful tools. With developments in next-generation sequencing, this is now being coupled to the whole genome amplification with the new enzymes and the new PCR protocols are being coupled with sequencing to do such things as look at the evolution of cells in tumors and to look at how the genetic defects evolve in those tumors. And I just show here two examples. In 2012, there's a very nice example looking at mutations and other gene defects in single cancer cells, but it's also been used recently to look at the process of meiosis in single human oocytes. And I can recommend that as a very interesting paper. So how are we using this technology then for pre-implantation genetic diagnosis? Well, actually, at the present time, we're stepping back and using a microarray-based technique, which is nevertheless extremely powerful. This allows us to sequence over hundreds of thousands of markers. These are called SNP markers, so-called single nucleotide polymorphism markers, and these are distributed right across the genome. So in a single test with a single array, we can genotype the markers across each chromosome, across all 24 chromosomes. So this is very powerful, and we can actually do this within 24 hours. So we can do this in time now, because we're obviously, one of the differences between 1990 and now is we have a little more time, as we'll see, because we can now successfully culture embryos to the blastocyst stage. So last year we published a very large multi-center validation trial of the technology. So this technology of genotyping these markers across the genome is used to genotype both parents and a sick child or another member of the family, and use this information to map whether an embryo has in fact inherited the condition. Now, I don't have time to go into this in any detail, but just really to show you here, and what you're looking at, this is chromosome 11 again in a beta thalassemia case, and for the child and for each embryo here, we have the paternal chromosome above the maternal chromosome, and the dots that you can see above and below the solid bars are actually the SNP markers. So if you think back to the previous practice where we had dozens of markers, we now have hundreds of markers or thousands of markers, and this forms a universal set that we can use for any single gene defect across the chromosomes. So in this case, beta globin is running here on the short arm, but if we wanted to look at the inheritance of anything else on chromosome 11, we could do that because we have markers automatically across the chromosome. And of course, if we wanted to HLA match in this patient, we could easily then just switch to chromosome 6, look at the HLA region, and decide whether in fact the embryo is matched. So in summary, carrier mapping is a breakthrough because a single universal test is applicable to almost any single gene defect. It's accurate and rapid and avoids the need for very time-consuming test development that's either patient or disease-specific. By far, the biggest application of pre-implantation genetics has become pre-implantation genetic screening, what has become known as pre-implantation genetic screening, i.e., some people would say PGD for aneuploidy. That is to say, we're looking to see whether the embryo has the right number of chromosomes or whether it has either inherited or has acquired an abnormal number of chromosomes. Now, in the vast majority of cases, in those situations, the genetic imbalance is such that there are only a very few exceptions that those embryos are not going to be viable. So it's thought to be very important, but it has been, as everyone in the IVF field knows, quite controversial. So this shows you the pregnancy rates, and we're all familiar with these graphs. This is actually the latest CDC information from 2012 in the US. And if we'd looked back at 1990, the graphs would actually have only come up to about 25%. So we're really doing very well. We've managed to increase rates to about 50% per embryo transfer, but we've done that at a cost, and particularly in the US, that comes at the cost of a very high level of multiple pregnancies, including many triplets, which of course are at high risk of obstetric complications and abnormalities at birth. And obviously we would all prefer to try and avoid that situation if we can. So one of the opportunities that these new technologies give us is actually to identify embryos which are abnormal and avoid transferring them, so that couples can elect to have a single embryo transfer with the confidence that they're transferring a normal embryo with a reasonable chance of pregnancy. So again, I think this will be familiar to many of you. Here again, we have the CDC information with the red line, which shows this steady and rapid decline until the point at which really we very rarely get a pregnancy in women of 45. If we superimpose on that the proportion of oocytes which are aneuploid, we see this steady increase. And this has been well established for decades, that there is this age-related increase in aneuploidies occurring during female meiosis. Now, many of these aneuploid embryos actually do develop, implant and develop successfully, but then they miscarry. So another consequence that also reduces live birth weights in older women is the fact that miscarriage rates come up very dramatically as you get into your 40s. So the PGS then is really aiming to tackle that issue. So this is going to be a real revolution, I think. So, as I said in my introduction, we've only had these genome-wide platforms for about 8 to 10 years. But over that period of time, we now have very solid evidence that there's actually an exceptionally high incidence of abnormalities. Some people would estimate that 50% of human embryos generated following IVF are aneuploid, and over 90% of those are completely non-viable, although some of them, a minority, will go on to generate a miscarriage, to cause a miscarriage. So this then, we've realized, is essentially the main cause of IVF implantation failure and pregnancy loss. And I think this is very significant. We really cannot ignore this any longer. And although PGS is very controversial because it's an expensive technique, at the moment it's still developing, there are errors and other complications, competing technologies, I don't think we can ignore it for very much longer. Most of these chromosome abnormalities actually arise in female meiosis, and I've already gone over that, and that is why they tend to come up with advancing maternal age, and still for reasons that we don't fully understand. But nevertheless, we've discovered that there is an exceptionally high incidence. I always believed that the incidence of aneuploidy in human eggs would come down essentially to zero. But in fact it doesn't, and recent studies, randomized studies, have shown really very high levels, even in women under 35 and on average around the age of 30. So it's a very significant problem across all maternal ages. And indeed, because of this age-related relationship, it's going to affect all women. But the difficulty for us is really that different women progress along this line at different rates. And it's impossible to say at what particular point a woman is, if a woman in her 30s has 30% or 2% or 0% at that time. So it's quite tricky. But what we do understand is that by the time we get to the age of 40 and above, not only do we have aneuploidies in the human oocytes, but we have multiple aneuploidies. And there's no question then that the genetic imbalance of these embryos is such that these are essentially not viable and they're simply not going to implant. And bearing in mind that women in their 40s, if they still have successful superovulation and egg collections of reasonable numbers of oocytes, will go on and have multiple treatment cycles, I don't think it's any longer acceptable, not at least to counsel them very carefully if they're not having any kind of screening. And finally, not only do we have this major source of abnormalities, but we have many other sources, including, of course, in some severe male infertility, we have elevated levels that are coming through from male meiosis and are present in sperm cells. But we also have abnormalities of fertilization, some of which get through. We check them for pronuclei. But in that validation study, 15% of embryos which had been scored as having 2 pronuclei were actually demonstrated in various ways to be abnormally fertilized. So there's really quite an exceptional level of aneuploidy. So applications for 24 chromosome screening. At the moment, this is an expensive and difficult technology, and so people are searching, trying to target this to the patients that are most likely to benefit. One very good target would be younger, good prognosis patients, and as we'll see, there's already been a randomized trial that shows there's a very significant benefit. And the idea here is to encourage them to have elective single embryo transfer, because these are the patients at particularly high risk of having those multiple pregnancies. So we really want to shift away from 2 and 3 blastocysts being transferred to a single euploid blastocyst if we can. Another group are the group in their mid-30s, which have a mid-range of abnormalities, in which we have a good chance of selecting euploid embryos and essentially avoiding multiple transfers and treatment cycles, but nevertheless giving them a significant benefit. We can't, I have to say, and I have to admit, we cannot change the quality of the eggs in any of these patients by testing them. But we can identify those which are normal, and we can give women very good advice about what their prospects are for pregnancy. No patient is an average patient. So the patient really wants to know, if I have this expensive IVF treatment, particularly without screening, what chance of pregnancy do I have? Is it 15%, 2%, or 30%, 40%? The only way of answering that at the moment really is to do this testing in order to be able to transfer normal embryos. And as we'll see, even in women in their 40s, if we can find that euploid blastocyst, implantation rates can be very good. And finally, for women in their 40s, obviously where the prospects are much less, nevertheless, as we'll see, there are a minority of women that are still producing many good eggs, and sometimes one or more of these are euploid. And of course, for them, it's of particular benefit to have those transferred ahead of the abnormal embryos, which might end up as a miscarriage. And then there are some other specific indications that I don't have time to go into, such as severe male factor that I've already talked about, or repeat pregnancy loss, and so on. And indeed, I and other people are already contemplating the possibility that in perhaps 5 or 10 years' time, and some clinics may start to do this in the very near future, should we consider screening all eggs and embryos simply because we should not be transferring abnormal embryos. Already we've had the first live birth, as we'll see, in 2009, following the introduction of the Array-CGH platform. And unbelievably, over that relatively short space of time, already we estimate that over 500,000 embryos have been screened using Array-CGH in approximately 200 labs worldwide. So there's been enormous progress. So in principle, 24 chromosome copy number analysis by array comparative genomic hybridization, or Array-CGH for short, is very straightforward. We take the single cell, or the small numbers of cells that we get from a blastocyst biopsy, in which we take perhaps 3 to 10 trophectoderm cells, and we amplify the DNA in those cells by whole genome amplification and then label it with a fluorochrome. We take control normal DNA and label that with a different fluorochrome, in this case green for the control DNA and red for the test DNA, and then co-hybridize that on DNA probes on a microscope slide. These DNA probes are spread across all 24 chromosomes. If the hybridization between the control and the test DNA is equal, then it tells us there's an equal number of copies in both. Whereas if the test DNA, for example, has a trisomy, then you'll get an excess of red hybridization. Or conversely, if there's a monosomy and we're missing a chromosome, then we'll get a depletion of the red and we'll get an excess of green. And the computer can pick this up with sensitive cameras, and the ratio will be shifted from this null point here. So that was the way the first arrays were done, but in fact the technology has moved on from there to so-called single-channel copy analysis. And the advantage here is that it uses fewer arrays, reduces the cost, and actually reduces the signal-to-noise ratio. So this gives us better, less noisy results down at the single-cell level. So we still have a certain amount of amplification bias, for example, that gives us noise from one probe to another. But the performance of single-channel analysis, which puts both of the control DNAs, one male and one female, on one array, and two now test DNAs on the other array, we can now, in silico, if you like, the computer can compare the hybridization of the green with the red here and vice versa. So the first pregnancies were actually achieved, and live births were achieved in 2009. And this is a nice picture of my friend and colleague, Simon Fischel, holding Oliver, who was the first baby to be born. And of course, this was an exceptional case in which this couple had had 13 previous failed IVF cycles. He was only looking at first polar bodies at that stage, and 7 out of 9 of them were abnormal. He nevertheless transferred the embryos that resulted from the other 2 oocytes, and Oliver was born. We now know that actually, because of the mechanisms of meiosis that I don't have time to go into in detail, that we need to analyze both first and second polar body to get an accurate idea of whether, in fact, an abnormality that we pick up in the polar body is going to translate into an abnormality in the fertilized oocyte. And what we see here is, in fact, an array CGH plot. And from the left to the right here, we have chromosomes 1 to 22, the X and the Y chromosome. And we have here the green line, which indicates this ratio from the array CGH. For all of these chromosomes, there's an equal amount of red and green. And so these are all euploid. They have the normal 2 copies. But here we can see there's very significant displacement, either up, showing that there's a gain, or below, showing that there's a loss. And particularly here in a magnified image, we can see that in the first polar body you can have a gain of a single chromatid, but then a loss in the second polar body, and no abnormality here, N for normal, in the oocyte. So we need to do both first and second polar body, and there's an ongoing randomized trial that ESHRE was sponsoring on polar body biopsy. What this also brings home to you is, in fact, there's not only a single aneuploidy in this oocyte and the resulting embryo, but several, all of which originated in female meiosis. Array CGH can be done on single blastomeres, and there are some examples here, including this wonderful slide that I like to show. And this is almost certainly a missed abnormality of fertilization, which has led to chaotic chromosome segregation, in which case you have random losses and gains across the whole karyotype. But people are moving very rapidly now onto Blastocyst biopsy, focusing this expensive technology on embryos which are likely to have the quality to develop well after transfer. And I'd just like to show some data of Nathan Treff and Richard Scott's group using a different technology. In this case, it's real-time quantitative PCR. But they've already examined a huge number of embryos. So this is a very important data for us to look at. And I've actually manipulated their graph to show you the proportion of blastocyst stage embryos which are euploid. And I think the staggering thing is that the average does not by any means come up to 100% in younger women. It plateaus off, and there's a huge number of aneuploid and essentially non-viable embryos. And of course, as women get older, the proportion of these aneuploid embryos gets greater and greater. So this is the kind of data that leads us to suggest that actually the proportion of abnormal embryos may be as high as 40 or 50% averaged across the board. But the remarkable thing is, and this is only a small retrospective study, is that if you can find, and this is blastocyst biopsy, PGS using array CGH, if you can find a euploid embryo, even in women of 41 or 42, then the implantation rates are really quite respectable and hardly drop off from the much younger age group. So I think many women in their 40s would really, be very excited about having this kind of implantation rate. But we have to bear in mind this is only a minority, very much a minority of patients, because most patients, when they get to this age range, you may find it very difficult to find even a single embryo that's euploid. When we look at the aneuploidy rates, there is this very high level. Even in the younger patients, you have 30%, and it comes up very sharply in women in their 40s. Consequently, because we're now screening out the aneuploid embryos, you'd expect miscarriages to decline. And we can see here that instead of that green line that we saw in the first graph, the rate of miscarriage, of course we can't eliminate miscarriage, not all miscarriage is associated with aneuploidy, does not increase even in this later age group. And in this case, actually, the number of patients in this group is very small, but there just happens to have been no miscarriages in this group. So we expect then, instead of having very high levels of miscarriage, to really improve miscarriage rates in that age group. So, of course, everybody understands that for these specific indications, what we need to be able to demonstrate is that the accuracy of our tests and the way that the embryos are biopsied actually results in better outcomes for patients. And so many groups have started randomized trials. This was one of the first with array CGH, in fact, in young, good prognosis women. I don't have time to go through all the details, and this was actually published in 2012. But essentially, this showed a very significant increase in clinical pregnancy rates per oocyte retrieval. But much more significantly, there were no multiple pregnancies in these women following single euploid blastocyst transfer. And we believe that this really is the future to encourage patients to reduce the numbers of embryos transferred so that we can reduce multiples and improve the health of children. But the level of aneuploidy in these young patients was really remarkably high, and in this case was actually 45%. Using other technologies, in this case real-time quantitative PCR, and this is again Nathan Treff and Richard Scott's group, they've done 2 randomized trials, small randomized trials, and they've done different things. In this one, it's comparing single embryo transfer with double blastocyst transfer. So the single embryo transfer is following blastocyst biopsy and screening by real-time quantitative PCR for aneuploidy versus unscreened double blastocyst transfer. And what they show is that these are equivalent in terms of clinical pregnancy rates, but of course in the screen group where you've only transferred a single embryo, there in fact were no multiple pregnancies. So again, a much better outcome for patients. And finally, they've looked at double embryo transfers to see what kind of increase they can get in pregnancy rates in a broader group of women. And in this case, the live birth rates following blastocyst biopsy and screening were essentially the same. However, already many labs are now moving on to the new technology, and the rapid advances in next-generation sequencing, I believe, over the next years are going to move us into a quite new territory. And so already there are protocols available to do 24 chromosome copy number analysis using the advances in next-generation sequencing, and I'm just going to go through that in principle here. So in this case, what we're doing is not trying to sequence the whole genome of the embryo, but simply to randomly amplify short sequences from throughout the genome of the embryo and map those back onto the different chromosomes, and then go back and count how many. Because this process is essentially random if you select the right regions of the genome, and that's where all the work comes in, of course, in developing these tests, we can get a very good readout of the copy number of the numbers of the target chromosomes that were originally present. So how do we do this? Well, so we run the sequencer to give us these short 36 base pair fragments, and these are mapped back to the human genome sequence. So we couldn't have done this, of course, years ago until we'd had the complete human genome sequence. We can then work out where these fragments are. In this case, this fragment is in this small section of chromosome 11 again. But if we zoom out and look for other fragments, we can find that also in this short region of this part of chromosome 11, we have 2 fragments. Now, if we'd had 3 chromosomes present instead of 2, we are more likely to have more fragments. And if we only had 1, we'd have less fragments to sequence. And so if we count them across all 24 chromosomes, we can get a very good readout. These are not proportional to chromosome size because we're looking at selected regions of each of the chromosomes that will give us a very nice linear readout. But what we can see here in this particular sample is that we've got double the number of mapped fragments in the test DNA in red, the embryo sample, versus our euploid control. And so we have this very nice linear relationship between copy number, chromosome copy number in the original sample, and the numbers of sequences that can be mapped to that particular chromosome. So if you compare the two, array-CGH and next-generation sequencing are highly concordant. And you can see here an example of 3 samples, A, B, and C, which have been run on array-CGH on the left-hand side. And here we've got a loss, a monosomy in this sample, and then run by NGS. And we can see that we can pick this up very well by using the NGS-based approach. But we have a larger displacement. And this gives many fewer ambiguous results, and we believe is a more robust approach and will give us improved outcomes. So again, because the relationship is linear, then we get this very nice relationship so that this essentially, if this is 2 copies, then you can see this is 3, and if there were 4 copies, this would have increased linearly with the copy number. And equally for loss, then you find here that this is equivalent to a single copy of that chromosome, in that case the X chromosome. So what are the advantages then of this new technology? Well, because we're only sequencing at very low read depth, obviously we still need to use whole genome amplification to get the DNA to sequence. We can do this very quickly now, so we can do this in the same time frame as array-CGH, It's almost 100% concordant with array-CGH, but we think in many circumstances it's more robust, will give less ambiguous results. It has a similar resolution, and we're still working on the exact resolution of the array-CGH, of the next generation sequencing. And we have this very nice linear relationship between displacement and copy number. Because the copy that we only sequence the low read depth, it's relatively cost effective to do this. And we can actually code the DNA in such a way that we can multiplex samples, which really reduces the cost. And indeed, using this kind of approach, the first birth has already been reported in 2013. One of the nice aspects, of course, is that in the future, not only will we be counting these sequences, but we'll also use the sequence information. And I believe that will happen quite rapidly. So very rapidly, I think we're going to be able to tell the parental origin of these abnormalities, for example, and we may be able to do many other things. And indeed, people are already wondering whether mitochondrial DNA is a good marker for embryo selection. And of course, we can sequence mitochondrial DNA by next generation sequencing as well. So there's a very big multi-center randomized trial that's now ongoing. It started in August of last year. And this is going to look at a very wide age range, maternal age range of women between 25 and 40. It's a multi-center trial which involves trophectoderm biopsy at the Blastocyst stage and single embryo transfer. All of the embryos initially will be vitrified. And obviously, I'm very excited to see how this works out. And we expect the results to be available within the next 1 to 2 years. So what's going to happen in the next 25 years? Well, of course, I can speculate as much as I want because I'm not going to be around for people to be able to say that that didn't happen or I got that wrong or whatever. Nevertheless, I'm going to have a go at talking about advances, some of which are already beginning to happen. I think these rapid advances in single-cell genomics are going to continue and give us the opportunity for increasingly sophisticated and cheaper technology to do these kinds of tests, and particularly by next-generation sequencing. I think the throughput is going to get better and better on cheaper and cheaper benchtop machines, and that will drive the cost down. In the last 10 years, we've got from millions of dollars in 2007 down to $1,000 a genome. I think that's going to continue to some extent, and so that's going to make this testing very much more straightforward. I think we're going to have equipment that will combine different tests. For example, high throughput next generation sequencing, but also a microarray scanning for such genetic applications. I think we're still going to need that for a period of time. And I think we'll have simplified protocols because some of this technology requires skilled molecular biologists still. But I think that's going to get simpler and simpler. And we're going to have lab automation. And I think eventually we're going to get to a point where the sample goes in one end and we wait and we get the answers out at the other end. And we may even have benchtop equipment that can be put into IVF clinics for certain things. And certainly, I think we're going to have targeted next-generation sequencing-based strategies that combine chromosome copy number, which is so important, I believe, for preimplantation embryos and preimplantation genetics, and sequence-level testing where it's necessary for PGD, so that we'll be able to combine PGS and PGD. They won't be separate tests any longer. So I think the power of genomics and next-generation sequencing then is going to, in fact, become integral to routine practice of IVF in the future. And not only screening embryos, but I think we're going to also be able to offer our patients a lot more screening of various kinds. So we'll be able to pick up karyotype abnormalities, of course, which are a frequent cause of infertility, but we'll also be able to pick up much more subtle copy number variants in patients that nevertheless can lead to infertility and other abnormalities of different kinds. We'll also be able to offer them things like carrier status for thousands of different conditions, including serious and many other forms of single gene defects. And of course, that's very important because even today, there are couples that have IVF treatment and nevertheless end up with a child. In my experience, for example, we had a couple who'd had IVF treatment, eventually succeeded with twins, who were both diagnosed with Batten's disease, and both died within a week of each other at the age of 4. We really want to be able to avoid that, and we'll have the technology in our hands to be able to do that, and it's not going to be expensive. The same kind of technology will allow us to look for fertility-related genes, and we're understanding more and more about that. And indeed, we'll have other advanced diagnostics. And finally, I think we will, essentially, I wouldn't have said this 15 years ago, I've changed my position on this, but I really believe, and I think some of the major centres are already beginning to think along these lines, that genetic testing of embryos will actually become integrated into routine practice. Because of the very high level of chromosomal abnormalities, I think this is going to become routine. We should not be transferring genetically imbalanced embryos. And once we do this and we have these very advanced technologies, whenever it's needed, we can then search for affected genes and avoid the transfer of embryos affected by inherited disease. So I think the next 25 years is going to be at least as exciting as the last 25 years. So in summary, by harnessing these exciting new advances in next-generation sequencing, which many of the major testing laboratories are already doing, we may be able to move from this to this, where we can offer significantly improved live birth rates to women in their late 30s and 40s, but also a reduced level of multiple pregnancies, and as we all want, healthier children following assisted conception. Thank you.